My name is Dr. Griffith. I am studying the two strains of bacteria that cause pneumonia. Of the two strains, one is coated in sugar and the other is not. This is the R and S strain. I am experimenting to determine which strain is harmful. First, I'm going to inject a mouse with normal R cell. It's alive. Next, I will inject a different mouse with normal S strain. It dies. Now I'm going to inject a mouse with dead S cells killed by heat. It's alive. Now I'm going to mix R cells with heat-killed S cells. Let the data reflect the mouse is dead. In conclusion, I've determined that the R cells are harmless and the S cells harmful. Also, during the last mouse injection, transformation occurred when R cells picked up genetic information from the heat-killed S cells that caused pneumonia. According to future scientists, this experiment will help determine that DNA is genetic material.
Hello, I'm Oswald Avery. I'm running an experiment to see what the factor is that kills those infected with pneumonia. First, I'm going to inject a mouse with R and heat-killed S cells and RNase, which kills RNA. He dies. Next, I will inject a different mouse with R and heat-killed S cells with protease, which kills protein. Let the data reflect that the mouse dies. And last, I will inject another mouse with R and heat-killed S cells and DNase, which kills DNA. And the mouse survives. This proves that the absence of DNA is what allowed the mouse to live, or that the DNA was the factor that originally killed the mouse. According to my scientific research, it was proven that DNA is genetic material.
Hello, I'm Alfred Hershey, and this is my colleague, Martha Chase.
We're trying to learn what transfers genetic material from a virus. Is it DNA or protein?
We will start by combining bacteriophage with 35S in liquid, then put it in this test tube. 35S is protein.
That's 35S. And this is bacteriophage. And here we have base liquid. Now we're going to mix all of these together and put them into a test tube. Look, a pellet of bacteria has formed at the bottom of the test tube. 35S, the liquid, and the virus are located at the top. And the pellet is bacteria.
Next, we will do the same thing but with 32P, which is DNA.
Here's the 32P, the bacteriophage, and more base liquid. Now we'll pour this into the other test tube.
In the test tube, you can see another bacteria pellet that formed and is made of 32P.
Now we can determine that the virus and the bacteriophage is in the pellet at the bottom of this test tube. From this, we can determine that DNA is transferred from the virus to the host.
These conclusions will help determine that DNA is genetic material.